Observe nature on a microscopic scale, and you see a relentless, chaotic engine at work. Organic matter mutates, adapts, and expands to survive in its environment through a process of trial and error spanning eons. For centuries, humans were restricted to the role of the botanist or the taxonomist, carefully observing and categorizing the results of this natural evolution without the means to direct it. This passive approach hits a wall when we face modern demands. Natural evolution hasn't produced an enzyme that breaks down industrial plastics in weeks, or a T-cell that can identify a specific novel synthetic pathogen. We are waiting for nature to invent solutions that it has no biological incentive to create. Reliance on natural selection is too slow for the pace of modern medicine. We have reached the limit of what we can achieve by merely reading the biological script. This limitation is forcing a shift in modern laboratories. The primary tool of discovery is moving away from the glass microscope and toward the computer terminal. Researchers are treating DNA as a modular programming language. They are mapping genetic sequences into distinct, repeatable blocks of code that can be edited and rearranged with mathematical precision. By treating genetic material as actionable software, biology becomes an engineering discipline. We are moving from studying how life works to designing how it should function. Building this new biology requires a workspace resembling an electrical engineering lab rather than a traditional hospital. Electrical engineers rely on standardized, interchangeable components like switches and logic gates to build complex hardware from simple parts. Biological engineers are adopting this exact framework through synthetic genetic circuits. An electrical AND gate needs two inputs to light a bulb. On the right, DNA acts as a biological gate, triggering a glowing protein when signals are present. These DNA parts snap together into circular instructions, then are packaged for deployment. Scientists now have the biochemical equivalent of a fully functional, compiled software program ready to be installed. We approach the living host cell as a blank hardware chassis, a vessel waiting for its operating instructions. The installation is physical. A microscopic needle pierces the cell membrane, injecting the synthetic code into the dark interior. The host cell's machinery immediately reads the newly inserted DNA and begins prioritizing these artificial instructions over its own natural programming. The output changes. Instead of ordinary metabolic functions, it manufactures specialized biofuels or secretes targeted medicines. The cell is a microscopic factory. It is a living machine executing human-written code. However, a silicon computer chip is sterile and predictable. A cell is wet, erratic, and governed by billions of years of competitive history. Biological code possesses a survival instinct that computer code lacks. Natural systems are built to mutate. Evolution favors organisms that adapt and change their internal makeup over generations to survive environmental stress. Over time, this natural drive toward mutation kicks in. The living cell loses pieces of our synthetic circuit or rearranges the code until the engineered program fails. The engineering challenge has shifted. We aren't just writing the code, we are fighting the cell's inherent drive for entropy. This friction between engineered precision and biological chaos is the current limit of the science. We face two possible paths. In one, the complexity of life consistently crashes our synthetic programs, keeping us bound to the slow pace of natural evolution. But in the other, we achieve a stable, rhythmic synchronization. The engineered cells divide and expand in a flawless, uniform pattern. If we can maintain this level of control, we will move past observing the world and begin dictating the biological script ourselves.